The last module in this chapter is module number 9. And in this module, we are going to discuss the brain, all parts of the brain. We're going to discuss the plethora of different structures we have in the brain and what those structures are responsible for as far as behavior is concerned. We are also going to talk about the various types of scanning techniques that we have in order to study the brain. What you may not be aware of is that we know so little about what the brain actually does. And that is because we have not been able to study the brain for really that long. Previous to 30, 40, 50 years ago, we were only able to study the brain when someone had died. In the last 20, 30, 40 years, we have scanning techniques, MRIs, PET scans, CT scans, that we use in order to study the brain of people who are still alive. And so there is a large portion of the brain that we don't even know what it does yet. But we do know parts of it, and that's what we're going to talk about in this chapter. So there are 4 questions that we are going to answer in this chapter. The first question, how do researchers identify the major parts and functions of the brain? In this question, we're going to talk about those various techniques, those scanning techniques, whether it is a CT scan, an MRI, a PET scan, things like that. The second question, what are the major parts of the brain? And for what behaviors is each part responsible? So we'll talk about each part of the brain, whether it's the 2 hemispheres, the 4 lobes, all the structures that are within those 4 lobes, and then the behaviors that each structure is responsible for. The third question, how do the 2 halves of the brain operate independently? Your brain is separated into 2 hemispheres, the left hemisphere and the right hemisphere. And each hemisphere is in control of various behaviors. And then the fourth and final question is, how can an understanding of the nervous system help us find ways to alleviate both disease and pain? Okay, so let's start out by talking about how we study the brain. How is it that we are able to study the structures and the functions of the brain? So it's almost like using a magnifying glass and spying in on what your brain is doing. And we do this by using various scanning techniques, and that's what we're going to discuss in these next couple of slides. One of the scanning techniques we have is an EEG. This is something that you may be familiar with, right? It's an electroencephalogram or an EEG. It records electrical activity in the brain, and it does so through electrodes that are placed outside of the skull. If you've ever lost consciousness, right? If you've ever been concussed, whether that was playing a sport or in an accident, and you are taken into the emergency room, the first thing the emergency room doctor is going to do is he or she is going to do an EEG, and that's going to tell them whether or not there is any electrical activity in your brain. If there is, then you're going to go further and find out where that is. If there is no activity in your brain, at that point in time, it's a very bad, serious accident that has occurred, and you are basically brain dead because there is no electrical activity in your brain. So an EEG will record electrical activity in the brain. Another type of scanning technique is a functional MRI or an fMRI. You may be familiar with the MRI scan. The functional MRI scan is used a lot of times in research, and that is only because your insurance company is not going to pay for an fMRI. It is extremely expensive to run the machine for an fMRI. Your insurance company would probably only approve a regular MRI. But the fMRI provides a detailed three-dimensional computer-generated image of brain structures and activity, and it does so by aiming a powerful magnetic field at the brain or other parts of the body. So the fMRI is just going to give you a very detailed 3D picture of the brain structures, and the activity of those structures. Another type of scanning technique is a PET scan. That's a positron emission tomography, or a PET scan. And this shows biochemical activity within the brain at any given moment. PET scans are used a lot of times when you're trying to determine what type of cancer someone has. If you had a biopsy and it came back positive for cancer, your oncologist will probably have a PET scan done to see what structure of the brain that cancer is actually affecting. That PET scan begins with an injection of a radioactive liquid. A fourth type of scanning technique is a TMS, or a transcranial magnetic stimulation. This shows electrical activity in a tiny region of the brain that is interrupted by this bombardment of a strong magnetic field, and researchers note the effects that that bombardment of magnetic field has on brain functioning. The TMS may be done in order to treat certain kinds of psychological disorders. And we'll talk more about that later on then too, once we talk about the various types of disorders and the treatments of those disorders later on in an upcoming chapter. So those are the four major scanning techniques, right? The four major scanning techniques: the EEG, the fMRI, the PET scan, and the TMS. We also have other types of, I don't know if you want to call them, I guess, scanning techniques. They just aren't of the more well-known major ones. But we have optogenetics, and this views individual circuits of specific neurons. So now you're getting very micro in discussing these neurons. You can also use a hydrogel embedding method, and this allows observation of individual brain cells and the wiring of brain activity. They talk about each of these in your chapter also. I think starting on page 71 and going through page 73, they'll give more details into each of these also. And the last one is the neuropixels. Now, the neuropixels are implanted probes that allow you to read activity in hundreds of neurons in multiple parts of the brain all at the same time. So they are implanted probes that allow neuroscientists specifically to read activity in hundreds of neurons in multiple parts of the brain at the same time. This picture is figure 1, and it is on page 72 in your textbook, and it just shows some of the various scanning techniques. So in EEG, this would show if there's any electrical activity in your brain. Remember, I said if you come in with a concussion, the first thing the emergency room doctor is going to do is they're going to do an EEG to see if there is any electrical activity. So that's the EEG. The fMRI, you see how this is a very detailed 3D look at various parts of the brain. The PET scan is in letter C. It displays the functioning of the brain at any given moment. And in this fourth one, in letter D, this is the TMS apparatus that they use. So the TMS, as it shows here below, it's the newest type of scan. And it produces this momentary disruption in an area of the brain. And it's going to show researchers then what activities are controlled by that area. In this chapter, or in this module, we are going to talk about both the old brain and the new brain. So here in the next couple of slides, I want to go back and I want to talk about the central core, or what we refer to as our old brain. So humans share some basic functions with more primitive animals. And these are directed towards an older, more primitive part of the brain. So if you think about some of our older, more primitive animals, like primates and monkeys, things like that, elephants, we share some of the more basic functions with those primitive animals with our older part of our brain. And this older part of our brain we refer to as the central core. It controls the basic functions such as eating and sleeping, and it is common to all vertebrates, not just humans. So this central core, this older brain, is the same for all types of vertebrate animals. It's in control of eating and sleeping, various functions like that. This picture is figure 2, and this is on page 73. And you can see how we have the central core, the old brain, which is at the very base of our brain. And then we have this newer, what we refer to as the newer brain, the cerebral cortex. And we're going to talk about that then here in just a moment. So the central core we share along with other primitive animals, a lot of our vertebrates. We as humans, though, stand out from some of those vertebrates because we have this new brain, or this cerebral cortex. So the central core, or our old brain. Let's start talking about some of the structures of the old brain. And remember, these we share with other vertebrates. So we start out with the hindbrain. Your brain is divided into the hindbrain, the midbrain, and the forebrain. The hindbrain is the very back of your head, and that contains the medulla, the pons, and the cerebellum. So the medulla controls a number of critical body functions, most importantly, breathing and heartbeat. So the medulla is extremely important for survival type of behaviors. Critical body functions such as breathing and heartbeat. The pons itself is a word that means bridge. So the pons bridges in the hindbrain, and it acts like a transmitter or a bridge of your motor information, which then coordinates muscles and integrating movement between the right and left halves of the body. So the pons, which means bridge, it's located in the hindbrain, and it transmits motor information between the muscles and movements between the right and left halves of the body. The pons is also involved in regulating sleep behavior. It's also involved in regulating sleep behavior. And then we also have the cerebellum. The cerebellum is the part of the brain that controls body balance. So the hindbrain, the back part of your brain, we have the medulla, the pons, and the cerebellum. Medulla in charge of critical body functions The pons is that bridge, coordinating muscles and integrating parts of movement between the right and left halves of your body. And in the cerebellum, which is very much involved in balance. Now, we also have the reticular formation. It's a part of the brain that extends from the medulla through the pons. And the reticular formation is in charge of changes in your level of arousal. So whenever you think about waking up and falling asleep, it is the reticular formation that is in charge of that. The reticular formation passes through the middle of the brain, which is now called the midbrain. Remember I said your brain is divided into the hindbrain, which is the back of the head, the midbrain, which is kind of like almost the top of your head, and in the forebrain, which is the front part of your brain. So the reticular formation passes through the middle part of the brain, called the midbrain, and it extends into the frontmost part of your brain, which is the forebrain. We also have the thalamus. The thalamus is the part of your brain that is located in the middle of the central core, and it acts primarily to relay information about the senses. So information about eating and drinking, taste, smell, touch, hearing, and sight. So it's relaying information coming in from those various senses. If we want to get more specific about the thalamus, I want to mention the hypothalamus. The hypothalamus is a very tiny part of your brain located just below the thalamus. And it is the hypothalamus that maintains homeostasis within your body. Homeostasis is this internal environment for the body. So it produces and regulates behavior that is critical to basic survival. So we think about things like eating and drinking and sexual behavior. The hypothalamus is in charge of those types of behaviors. Figure 3, this is on page 73 in your textbook, and this figure is just going to show you where these various parts of your body or of your brain are located, right? So here we have our spinal cord, and here we're talking now about our hindbrain. So we have the medulla, right? We have the pons, we have the reticular formation, and the cerebellum. Now we move into the midbrain. We have the thalamus, right? We have the hypothalamus. We have the pituitary gland that we talked about before, which is in charge of hormones. And then as you'll see here in just a little bit, the cerebral cortex and the corpus callosum. And we'll talk a little bit about that coming up in just a bit. Okay, so now let's move on, still in the central core that we're talking about, the old brain, but let's move on to the limbic system. The limbic system is kind of like the main emotional center of your brain. So it controls all of your kind of emotion and motivation types of behaviors like eating, aggression, and reproduction. The functions related to emotions and self-preservation. The two main structures of the limbic system are the amygdala and the hippocampus. The limbic system and hippocampus in particular play an important role with both learning and memory. You may have heard in a previous course about the gentleman referred to as HM. HM was in a bicycle accident and had damage done to his hippocampus, and after the accident he could not form any new memories. So he had some of his long-term memories, but he had no new short-term memories that he could remember or that he could form. And because of the limbic system structures and functions, because they are so similar to those of other mammals, the limbic system is sometimes referred to as the animal brain. So a lot of your primates, a lot of your other vertebrate types of animals have the same brain structure in the hippocampus or the limbic system. This picture is figure 4, and you can see this picture in your textbook on page 74. And again, it just shows you where the amygdala is. The amygdala is like the main emotional center of your brain. I know I said that the limbic system overall was emotional, but the amygdala is definitely the main emotional signal or the main emotional center of your brain. It shows you where the hippocampus is, and then in relation to the spinal cord. Now, everything that we've talked about so far was the old brain. If we move on now, we talk about the cerebral cortex, which is our new brain. And if you remember the picture in the slide, a couple of slides previously, we had that picture of both the old brain and the new brain. And we said that the cerebral cortex is also referred to as the new brain. And it is responsible for the most sophisticated information processing that occurs in the brain. One of the main characteristics, I guess you could say, of the cerebral cortex is the lobes. And we have 4 major sections of the cerebral cortex that we refer to as the lobes. One of those is the frontal lobes, and this is the front center of the cortex. We'll talk about more about these lobes later on. The parietal lobes is behind the frontal lobes. The temporal lobes are in the lower center portion of the cortex, and then the occipital lobes are behind the temporal lobes. So the cerebral cortex is divided into 4 lobes: the frontal lobes, the parietal lobes, the temporal lobes, and the occipital lobes. These 4 lobes are separated by deep grooves, which are called sulci. Now, figure 5, this is on page 76 in your textbook, and here we can see your 4 lobes and then the various structures within those. So we have the frontal lobe, that's the motor area and Broca's area, which we'll refer to in just a minute. We have the parietal lobes, which is the somatosensory area and the somatosensory association area, again, which we will come back and talk about. The temporal lobe, which is your main auditory cortex, also Wernicke's area and the auditory association area, and then the occipital lobe, which contains your main visual cortex, the visual area, and the visual association area. So let's talk a little bit more in depth about each of these lobes and the structures within the area, or the structures within the cerebral cortex. So we're going to start out with that frontal lobe and the motor area of the cortex. It is largely responsible for all of your body's voluntary movements. So again, voluntary versus involuntary. Voluntary meaning those things which you are in control of, which you voluntarily do. And it's the motor area that is responsible for that. Every portion of the motor area corresponds to a specific locale within the body. Whether it is your fingertips or your arms or your legs or your head or your neck or your back or whatever it is, but every portion of that motor area corresponds to a specific locale within the body. Researchers, specifically neuroscientists, have identified the amount and relative location of cortical tissue that is used to produce movement in specific parts of the human body. So that is the motor area of the frontal lobe. Now let's move on to sensory areas. The sensory area of the cortex. So now we're talking mainly about the parietal lobe. The sensory area is the site in the brain where the tissue that corresponds to each of the senses, with the degree of sensitivity related to the amount of tissue that is given. So your senses, such as vision and hearing and taste and smell and touch. There is a specific area in the brain for each of those senses, and the amount of tissue that's given to those areas, they are seen as more sensitive. So, for example, the somatosensory area in the parietal lobe encompasses specific locations associated with your ability to perceive touch and pressure in a particular area of the body. Whether it is your toes or your thighs or your calves or your forearm, whatever area it is, right? Whether it's touch and pressure to that specific area of the body. The auditory area is in the temporal lobe, and that's responsible for your sense of hearing. And then, as I mentioned on the previous slide, the visual area is located in the occipital lobe. So we have the frontal lobe, which we said deals with motor behavior. The somatosensory area deals with touch and pressure. The auditory area of the temporal lobe is your hearing senses, and the visual area of the occipital lobe is your sense of vision. Now, whenever I said, right, let's go back and think about this. The sensory area, it says the site in the brain of the tissue that corresponds to each of the senses with the degree of sensitivity related to the amount of tissue in that area. I reiterate that because we have certain places in our brain, in our sensory area, that have more tissue than others. That means those places are more sensitive than others. And if we were going to show that on a person, it would look like the figure in the next slide. So this is figure 6 on page 77. And it really looks somewhat strange, right? But you can see that your hands and your fingertips, your lips, your tongue, your nose, those look overly large in this figure, in figure 6. And that's because those areas have extra tissue, which means that they are overly sensitive. And that makes sense. If you think about it, from a survival type of standpoint, you use your hands to touch a lot of things. So it's going to protect you if something is hot or if something is sharp. The same with your lips and your tongue. You're not going to put something really extremely hot in your mouth, and you're not going to put something sharp into your mouth. So it makes sense that this somatosensory area shows those places on your body which are extra sensitive, meaning that they have more tissue on them in your brain. Next is the association areas of the cortex. Previously, we had mentioned the visual association area, the auditory association area. What we're talking about by saying an association area is that it's one of the major regions of the cerebral cortex, the site of the higher mental processes such as language and memory and speech. So a really well-known example is the case of Phineas Gage. And they talk a little bit about Phineas Gage on page 78 in your textbook. Phineas Gage, we're talking about the late 1800s. He was a railroad foreman in Vermont. And because he was a foreman, he was in charge of a lot of different things like planning and organizing and everything that goes along with being a foreman for a group of men. One day, whenever they were out working on the railroad, there was an accident accident in one of the railroad ties. There was an explosion, and the railroad tie went up through Phineas Gage's cheek and out through his brain. And it went right through the frontal lobe area of his brain. And after this accident, Phineas Gage went on to live for 12 more years, which is just amazing in and of itself, that in that point in time that he went on to live for 12 years. But he pretty much lived in his body and not really in his right mind. There was such a change in Phineas Gage's personality after the accident compared to how he was before the accident. He did not have control of his emotions. He wasn't good with organizing. His speech really went downhill. He could never again become a foreman. Injuries to the association areas can produce what we call aphasia. Aphasia is where you have problems with language. And there are two specific areas in your brain, Broca's area and Wernicke's area. One is responsible for understanding spoken language, and one is responsible for making spoken language. So both of these areas, Broca's and Wernicke's, involve spoken language. Now, if you have aphasia to one of these areas, that means that you have difficulty with spoken language. So with Broca's aphasia, you have difficulty speaking. You understand what is being said to you, but you cannot answer correctly. Now, it is not as though this person is not able to live in society, live normally, because what they could do is they can understand if someone says, "What time is it?" You just cannot answer them. But what you could do is you could write it down, because you have no trouble understanding what was said to you. You just cannot answer. The opposite of this is if you have Wernicke's aphasia. This is where you have difficulty understanding what other people are saying to you. So if I would say to you, "What time is it?" and you have Wernicke's aphasia, you have no idea what I asked you. You may say, "The sky is blue. I think it's really cold outside." You have no trouble speaking, but you don't understand what someone is saying to you. Now, what that other person could do is they could ask you, "What time is it?" And you would say, "It's 7:15. So if someone has an aphasia in their brain, it doesn't mean that they cannot go on and live a normal life. They just have to learn to adapt. Students always ask me, well, do they gain this back? Do they get it back? It really depends on how severe the brain injury is. Some patients who have a stroke, that stroke may affect either Broca's area or Wernicke's area. So they may have to relearn how to talk again. They may have to relearn language to understand what is being asked of them. But it really depends on whether they gain this back or not. It depends on how severe that damage is. Whether you had a stroke, whether you were in a car accident, or you had a head injury, or whatever the situation may be. Let's talk a little bit about neuroplasticity and the brain. Neuroplasticity is your brain's ability to change throughout your lifespan. And this is through the addition of new neurons and new interconnections between those neurons, and the organization or reorganization of information processing. You will continue to develop new neurons throughout your lifetime until the day that you die. However, that process really slows down after, I would say probably after the first 6 years of life, and then really after your teenage years, you really start to see a slowdown. And it will continue to slow down throughout your lifetime. You will still continue to develop new neurons, but it is at a much slower pace. So if you do things to kill off those neurons at a faster pace, whether that means that you're doing drugs or you're drinking alcohol or you're listening to really loud music. Those things continue to kill neurons. You're still getting new neurons to replace them, but it is at a much, much slower rate. Neurogenesis is the creation of new neurons, and it is now known to occur in certain areas of the brain in adulthood. Let's come back around now and let's talk about, we've just talked about, the four lobes, we talked about the association areas, we talked about all these structures in your brain. I want to come back now and I want to look at your brain overall, and I want to discuss the two hemispheres of your brain. So the brain itself, as you can see here in the center, in this like little flow chart, so to speak, is that the brain is divided into roughly two mirror images of themselves. These are referred to as hemispheres. They are symmetrical left and right halves of the brain, and they control the opposite side of the body. So your left hemisphere controls behaviors on your right side of your body, and your right hemisphere controls behaviors on the left side of your body. The left hemisphere tends to process information, particularly the verbal areas. The right hemisphere processes information globally, particularly the nonverbal areas. Okay? So your brain is divided into these two halves, the left and right hemisphere. They control the opposite sides of the body, and it is the left hemisphere that's in charge of verbal areas of your brain, those that control language, and the right hemisphere is involved in nonverbal areas. We use this term lateralization. It's the dominance of one hemisphere of the brain in specific functions, such as language. The differences between the two hemispheres are not great, and the two hemispheres do work interdependently. Now, they can communicate with each other by way of the corpus callosum. So if information comes into your right visual field, it would go to your left hemisphere. If information would come into your left visual field, it would cross over to your right hemisphere. And we just talked about how the hemispheres are in control of different behaviors. And I'm bringing this up because in the next slide, we're going to talk about patients who have had what is known as a split brain surgery. So previously, we're talking many, many years ago, we do not do split brain surgeries anymore. But for patients who had very severe seizure activity, before we had drugs to try and control the seizure activity, in very severe cases, surgeons would go in and they would sever the corpus callosum. This did not cure the seizure activity, but it would keep the seizure activity to one side of the brain so that it wasn't as severe. Again, severing the corpus callosum does not cure seizure activity, it just kept it to one side of the brain. However, because the corpus callosum is severed, now the two hemispheres cannot communicate with each other. So in split brain patients, let's just reiterate, the corpus callosum has been cut or injured. And again, this was done as a last resort for severe epilepsy. So the studies that were done on these split brain patients, the studies of how each hemisphere operates, they showed that they should be regarded as different. The two hemispheres should be regarded as different in terms of the efficiency with which they process certain kinds of information. In this picture, this is the last slide in this module. This is figure 7, and this can be found on page 83 in your textbook. And it shows the hemispheres of the brain. If you see in letter A, the site where the corpus callosum is severed, right, that is right here. and this is the corpus callosum, the right cerebral hemisphere, is now separate from the left cerebral hemisphere. And they did research with patients where they would have them sit at a table and there would be a screen in front of them so that they could not see what was on the other side of the screen. But they would ask them to reach around and feel what was on the other side of the screen. So if you look in your textbook, I don't have it here, the description in front of you, but it says a split brain patient is tested by touching objects behind a screen. Patients could name the objects they touched with their right hand, but they couldn't name them when they touched them with their left hand. Now think about this. Remember we said that the left hemisphere is in charge of verbal behavior and the right hemisphere is in charge of nonverbal behavior. And remember, information crosses to the opposite side of the body. So if you touch something with your right hand, it's going to go to the left hemisphere, and then you could say what that object was. However, if you touch that object with your left hand, it's going to go to the right hemisphere, and you cannot say what the object is. In a normal patient who does not have a split brain, the left and right hemispheres would just communicate with each other, and they would then say, I see a piece, or I have a piece of chalk, or this is a hairbrush, because the left and right hemispheres can communicate with each other. But for a split brain patient, this does not always work. Okay, that is the end of Chapter 3 on neuroscience and behavior.